Introduce our first speaker. Today we have Soumesh Padhi. He's received his MS and PhD in biotechnology from Utkal University in India. His PhD research focused on understanding the diversity of heterotrophic bacteria and their application in freshwater fish culture systems using biofloc. He then worked as a research associate on the effect of nanoparticles on fish growth when used as a feed additive. And for the last couple of years, he's been working in my lab at the University of Minnesota on molecular diagnostic development, and he'll present today on his work with virus discovery. So take it away, Soumesh.
Thank you, Nick. Welcome all. And as Nick mentioned, I am from India. My state is southeastern part of India. I did my PhD research work at the Central Institute of Freshwater Aquaculture, which comes under Indian Council of Agricultural Research Organization. I joined Minnesota Aquatic Invasive Species Research Center in 2017 September, and I am working with very dynamic research personnel, Dr. Nick Phelps, Dr. Sunil Kumar Moore, Dr. Alex Primus, and our bioinformatician, Dr. Vikas Singh, and Isaiah Tolo, who is under the process of getting the doctorate. So my today's my topic is biome diversity in North American freshwater fish mortality events. So starting with my work, like all our research group, we are generally focused on the discovering of novel fish viruses in the different fish species. So based on this idea, we started this research work. Couple of years ago, our research team got very a large number of natural occurring fish mortality event samples. So we thought how we can use those samples for our research community. So we, based on those idea, we have three things to consider. First, why we need to see the use the fish mortality events or fish sample for looking for viruses. As we all knew that fish has very ancient evolutionary origin dated back to the 540 million years ago. And as a pathogen, the viruses and the host, they can reciprocally affect each other's evolution. They are co-evolute during the evolution procedure. And especially for pathogen as a virus, it is also known as very specific to a particular host. And next idea was what we are going to look out there in the samples. So first, what type of virus is present in the samples, and to explore largely unexplored fish viral communities. So how we are going to achieve those kind of things, like, so first, the recent advanced method is the next generation sequencing. So the viral metagenomics comes in the picture. So the advantage of doing viral metagenomics is, like, for virus, we don't need to have a new cell culture system to cultivate the virus, or we don't need any conjugate gene to categorize the different viruses. And how this data will going to help the other community is, like, we can determine what are the emerging viruses present in all those samples. So ultimately, the final aim is this data will be the baseline data for the fish pathologists to use as a diagnostic tools. So starting from my research or experiment outline, we recently, means last couple of years, we as I mentioned previously, we got a large number of natural occurring fish mortality events, and our sample consists of 7 different fish species, 7 different fish families, which has 13 different fish species. And all those data represent 15 naturally fish kill samples. And the interesting thing is here, like, we got a large number of common carp-specific mortality events throughout the last throughout the couple of years. So that's why we thought, let's include the healthy common carp and see what are the virus population is different and viral diversity present in the common carp specific mortality. So we have included, we have collected healthy common carp from 5 different lakes, and also we have included the silver carp and collected from the river Illinois, from the Illinois. And that included, we have total 59 different geographical location samples, and all those samples also represent 5 different Midwestern states of United States of America, like Minnesota, Wisconsin, Iowa, Pennsylvania, and Illinois. So majority of our sample belongs to the Minnesotan state, and all are from the natural occurring fish mortality events. So all these samples, we proceed for the next generation sequencing. The first step of next generation sequencing was the sample collection. So as all of us, we know that natural fish mortality events is very random events, and we relied all our sample collection on the government agency, different state government agency, and the fish health enthusiastic person, and normal civilian person who used to report the normal fish kill events through our website. Once we get the report, usually we went there to collect the samples, or sometimes samples mailed us. So once we got the sample, we tried our utmost care to do the necropsy in aseptic condition, and we tried to collect, our best attempt was to collect 4 different tissues like brain, gill, kidney, and spleen. Once we collect this sample or organs, we pool them according to the species or organ type or different geographical location. For example, if we collect if we get 8 different bluegill samples, and we do the necropsy and pool the specific like brain sample are pooled into one group, and based on that, we were end up getting 216 samples. So all the samples were homogenized through tissue homogenizer, and all the samples were treated with DNAs and RNAs to remove the degraded nucleic acid. And the total nucleic acid was extracted using Quiamp mini-elute spin column kit. And then the nucleic acid were QC'd, and the concentration were checked and processed for the library preparation. And the sequencing was done on the HiSeq 2500 sequencing platform, and the chemistry were used 125 parent sequencing. This generate a huge number of sequencing data. To analyze those data, we have developed one in-house bioinformatics pipeline to detect the raw reads and filter those raw reads to get the good sequence quality sequence read, and then we use those read to remove the host genome, and the remaining sequences we used for de novo assembly. Once the de novo assembly formed the context, we searched for the NCBI database for viral hits, and then all those reads or all those contexts were annotated for viral context. And again, to get the abundance of what type of or what is the population size of virals present throughout in our data, we use those annotated context and map to the filter of FASTQ reads. And here I want to mention, like from all our data, only 10.8% reads were showing some hit to the viral genome or viral sequences. So describe in overall figure, all those from the 10.8% viral reads. From there, mostly the 85% reads belongs to the bacteriophage, and 9.9, around 10% belongs to unclassified virus, which do not have any sequences or any taxa present in the online database. And from there, and 5.8% reads or contexts were classified as the classified viruses. Again, we went in depth to classify those classified viruses. We saw like the single standard DNA viruses are more abundantly present, around 60%, and the double standard viruses are around 24%, and the least viral DNA viral sequences are from the single standard negative sense RNA. So again, we went through in depth and analyzed what type of viruses are present, even though those are single standard or double standard. So in total, the DNA viruses, we found total 25 families are present, and among them, circoviridae is most abundant, which is a single standard DNA virus. And we also found the alloherpes virus, which also contain the koi herpes virus, and the koi herpes virus is specific to the common carp. And we also found large DNA viruses like poxviridae, iridoviridae. And the red marking here is showing that those family contain the pathogen or the virus which can infect the vertebrates. And the right hand side, you can see the bacteriophage, and we detected total 14 bacteriophage families, and among them, Podoviridae was the most abundantly present. And here I want to show you something like unclassified Caudovirales. This is an order. There are a lot of sequences which we didn't able to or classified which Caudovirale families those belong to. So similarly, for RNA viruses, we analyze in detail. Like in RNA viruses, we discovered total 29 families present in our data, and out of those, picornaviruses and reoviruses are viridae are abundantly present. And also you can here see there are a lot of sequences which belong to the unclassified picornavirales order. So they are pretty novel. We really don't know whether which class those belong to. So here also the red marking shows that those families also known for their infecting in vertebrates, infecting vertebrates. So next we went to do some detailed analysis. So first we took, for example, as a picornaviruses. Here we want to see how the picornaviruses are divergent or how similar picornaviruses present in our data. So the first criteria was those sequences or those contigs we need to take which have the 80% amino acid level, lower than 80% amino acid level similarity, and the sequence or contig size should be more than 3000 base pair. So on the left hand side here, you can see this is the consensus identity, and these are the different fish name, and the number here are the different context number. So out of those, we found that there is one large virus present in the common carp, which has the genome around 12,000 base pair. Normally, the reported genome, the picornavirus is vary within 8,000 base pair to 9,000 base pair, but still there are some recent report of picornavirus present which are more than 12,000 base pair. So again, all these sequences contain both RNA-dependent DNA polymerase, structural and non-structural genes of the viruses. So next we went to categorize the reoviruses. So here we just took the RNA-dependent RNA polymerase to get the phylogenetic evolution, phylogenetic tree to see the evolution of reoviruses, what we found in our data. Interestingly, we found two reoviruses, which is almost complete RNA RDRP gene. So we tried to prepare one phylogenetic tree. So here you can see those two contexts are pretty divergent to any reported reoviruses. And if you look for the scale, it is totally very divergent. On the other side here, you can see the cadipoviruses, which also a recently discovered and recently discussed viruses belong to the reoviridae. We found three different RDRP gene in the yellow perch, common carp, and bluegill samples, and they are two very divergent with respect to reported any cadipoviruses. So next one was the black Crappie Pox-Novel viruses. This is very interesting in our data. So we found one specific pox virus present only in the black Crappie sample. And the interesting stuff here is like all the hits are from the gill tissue. And we had a total 8 black Crappie specific mortality events. So all those samples contain black Crappie pox viruses. So in our data, we were able to generate 181,000, around 81,900 base pair of sequences for this specific to the black Crappie pox viruses, but we were not able to complete the whole genome. So now, based on those sequences, we try to find out any ORF present in our data or not. So our criteria was like the find out the particular ORF which has the minimum length should be 504 base pair. So ultimately, we end up with getting 180 ORFs in the novel pox viruses. So again, we just tried to see how divergent this pox virus is based on the core protein gene in the pox virus, that is P4A, that is also used as some research used as a conjugate gene to categorize or to see the evolutionary pattern of a particular pox virus. So here you can see only the only this black Crappie is showing similarity to the salmon gill pox virus, which is the only reported pox virus till now. And the right hand side, you can see different genes from the salmon gill pox virus, how they are showing amino acid level similarity to the recently we discovered black Crappie samples. And most of all the genes are below 80% amino acid level similar, means below 80% amino acid level similarity. It is clearly defined as the black Crappie what we got in our sample is totally divergent and totally new virus we have detected in our sample. So there are a few other interesting findings in our data I want to mention here. Like in the total data, total 84% of viral contexts are below 80% of amino acid identity to any reported viruses present in the database. So this signifies how divergent, how novel our fish viruses are. And there are a few other stuff I want to show you here, like we also detect some parvoviruses in goldfish and common carp. So one of the parvoviruses in goldfish, which is showing 39% amino acid similarity to the human oncogenic parvoviruses, and the length was 3031 base pair. In common carp, also we detected another parvovirus, which is showing 31% similarity to the virus which can cause diarrhea in human. And another stuff we found in the yellow perch was novel rhabdovirus in the spleen tissue only. So that that contact is showing 45%, less than 45% similarity to the nucleoprotein and L protein of an insect rhabdovirus. So again, the sarcoviruses, as I previously mentioned to all of you and show you, like our data is dominantly present with the sarcoviridae. So we all know that sarcoviruses has a genome from 1.8 to 3.8 kb. So our criteria was like just select those sequences which has more than 1,000 base pair. So we found out that our sample has 22 to 80% of amino acid similarity to any known sarcoviruses. The right hand side, you can see the common carp has more abundant in the sarcoviruses. So next thing is, apart from this experiment, we also did some whole genome sequencing of koi herpes virus and carp edema virus. So recently we have isolated koi herpes virus from the wild common carp of Minnesota, and we were able to assemble the whole genome. This koi herpes virus is showing similarity to 99.8% to the KHV isolated in the Poland. And using Oxford nanopore sequencing, we were able to assemble partial genome of carp edema virus. Here you can see how big the context size is. So next, the future direction and we first think is most of the sample, the DNA viruses, we were not able to assemble the whole genome. So that's why we thought let's do the long read sequencing. Recently, we just finished the Oxford nanopore sequencing from total 58 pools, and it has generated total 3 TB data. And apart from that, we were just doing another. We will be comparing this Oxford nanopore sequencing data with HiSeq data to get the whole genome, and apart from that, we will figure out which method is best to detect rapid diagnostic of any fish mortality events. So all we know that any good opportunity comes with its own challenges, unique challenges. So throughout our experiment, we faced a lot of challenges. So here, the first thing is the sample quality. All our sample, most of the sample are from the fish mortality events, and we didn't have any control over the sample, what we are getting. So the main disadvantage of NGS is we need to put a very good quality of tissues or initial material, but we didn't have that opportunity. So the next thing is host or bacterial contamination in our sequencing. Even though we treated our sample with DNAs and RNAs, but we still got 90% of the sequence reads belongs to host or bacterial reads. The sequencing depth, that is also in future, we will be doing more sequencing depth so that we'll get per sample, so that we'll get more reads per samples. And the detection of fish virus is totally hard because there is very few data available throughout online data available, and it is pretty hard to annotate and figure it out whether which families or how these viruses are belong to. The next challenge we faced, like bacteriophage, although all the samples we virus reads are characterized virus read, there are some novel viruses, but most of the around 85% of reads belongs to bacteriophage. That we think these are all the sample from the fish mortality events. That might be the cause. That's why we got a lot of bacterial phage, because sample may be degraded. And another challenge we face, like insects, plant, or environmental viruses. So this is again another challenge. So we cannot really figure it out whether this is belongs to those insects or plant, or it is really divergent to the divergent viruses, because there is no report on the fish viruses. So again, the next challenge, it is a huge data. So analysis is still going on, and we'll be updating all of you, of our results. So by this, I want to thank you, all of you, for your patience, and I want to acknowledge Veterinary Diagnostic Lab, University of Minnesota, for helping this work done in their laboratory, and the Minnesota Department of Natural Resources, Fish Health Management person from Minnesota, Wisconsin, Iowa, Illinois, and Pennsylvania, and all the persons who have contributed in the sample collection. Thank you. I am open to suggestion or any discussion. Thanks.
Thank you very much, Soumesh. Just a reminder, if you have questions for the presenter, you can unmute yourself or type it in the chat box and I can read it to them.
Okay.
We have about 4 minutes.
Hey, Soumesh, I'll get started with the questions.
Yeah,
great
please.
talk. You know, this is Esteban.
Hi, Esteban.
How are you doing, man?
I'm good. Thank you.
They've been keeping you busy, so that's great. So Soumesh, going back to the pox virus finding that you have, and you mentioned and you present only the Salmonid pox virus. I don't know if you can go back to that slide. And I'm just curious because, and you can educate myself into where this is falling now, but I thought that Carpedema virus was also considered to be a pox virus. So you have a bunch of carpedema virus, hits in the samples that you presented, and even I don't know if you were able to assemble the whole genome of carpedema virus already, but I'm curious, in one of the genes that are used also to classify carpedema virus is that P4, the P4A. So I am curious as to where does it fall in here, if you were putting the carpedema virus in this tree, because again you have already those sequences. So I'm just curious why you didn't put them in there.
So yeah, that's a nice question. So I didn't annotate those carpedema virus sequences what I got, but this black crappie sequence P4A gene, that's why I just took out. And carpedema virus, I think based on our data, it's the P4A gene. Only the short fragment are reported online, and based on those data, I took the P4A gene around 500 base pair and initially matched with the Salmon Gillpox virus. It was showing around 82 or 85% similarity. But if you see, yeah, I didn't include those data here, but if you see the black crappie one, the P4A gene is totally divergent. It is below 80%. So maybe, yeah.
Yeah, that's exactly why I'm asking about the carpedema virus, just to see where does it fall.
Yeah.
If you compare it, because I think even for carpedema virus, there has been some genetic diversity even shown within the P4A, and some people have called them different genetic groups. So I'm just curious, where does it fall? Because you have carpedema virus in those lakes in Minnesota. So yeah. I don't know if if it will fall closer to the gill or to the black crappie one.
Yeah, I'll next time. Yeah, I'll include those part. I didn't have the annotated those carpedema virus genome, so I didn't include that one.
Yeah, and I know that carpedema virus is still some people discussing if it is a true pox virus or not. So I was just curious if it was that it changed or somehow that I didn't know about it. But yeah, just curious about it. Thank you.
Yeah. Thank you. Nice question.
Soumesh, that was a really great talk. This is Jan.
Thank you.
You mentioned that a lot of the samples were degraded, so probably you weren't able to find out what the actual cause of the mortality was. Was there ever any attempt to try to figure out what the cause of mortalities were in those fish kills?
We just figured it out in the common car, but not in all different species. Our aim here is to see what type of virus population present. We didn't look for is these viruses are those viruses causing any pathogen or not. We are just looking for what type of virus present. So.
Okay. Cool. And then also, I noticed that there's very low diversity in RNA viruses in there. And then just seeing that there was a lot of degradation of the samples, do you think that that could be an artifact, that some of the samples were so degraded and RNA viruses maybe are susceptible to decomposition, or that you wouldn't be able to detect them in sequencing?
So there's the challenge we found. Yes, that is another factor, maybe the degradation of the sample. Well, that's why we didn't get a very good sequencing result. And yeah, that's another challenge too. Another factor, yes.
Great, thanks. Great job.
Thank you.